All the data which I will present to you come from the ITT population. We had 2 early discontinuations in each arm. The 2 on asthma are shown here. 1 terminated due to asthma exacerbation and 1 due to consent withdrawn. The 2 discontinuations of active were due to adverse events. 1 individual reported moderate flu-like symptoms 3 days after the second injection and terminated, and 1 individual had actually emotional distress. We actually don't think that this has something to do with our study drug, but rather with the fact that this person was part in a clinical trial and probably she shouldn't have been there to start with. It was also reported that she had tachycardia, but that was self-resolving. Actually, all the side effects in the study were self-resolving within a short period of time. Now, the first clinical endpoint was that symptom and medication score. Patients were distributed an electronic diary where they did, over the 12-week period of the study, report how much symptoms they had. They could give to the symptoms 0 to 4 points, depending on whether they had no asthma symptoms or very bad asthma. Same for nighttime symptoms, 0 to 4 points. And then our patients had access to a short-acting beta-2 agonist as relief medication, and depending on the number of puffs somebody took, he would get 0 to 6 points. And then the combined score is simply the average of the sum of the night and daytime symptoms and the medication score. The result on this endpoint is shown on this slide here. I would like to draw your attention to the orange curve first, which are the placebo people. What you see is that during the 4 weeks when they were on the 100% corticosteroid dose, that they were indeed stable, so they did not change. But when the corticosteroid was reduced to 50%, they worsened. So the higher the score here, the worse the disease is. And then when they went to 0% corticosteroid, they did even worse. If you go into the blue line, you see the active ones. Those improved already after 1 injection of the study drug, and after the second injection of the study drug did we see the first statistically significant difference between active and placebo. And then again from week 6 onward, at every time point, were they highly significantly better than the placebo patients. Also, and you will see this throughout the presentations, these patients at the end of the trial did better than at the beginning of the study when they were under the standard corticosteroid therapy. So I would remind you here, they have stopped taking inhaled corticosteroids and they did better under our therapy than they did at the beginning of the study. The combined score is, as I said, the combination of the symptom and medication score. I would like to show you here how these subscores developed over time. It's the same picture again. Overall, a highly significant difference between active and placebo. In the repeated measures analysis, we had the same here, also in the combined score. In the symptom score, we found that the actively treated patients had 30% less symptoms at the end of the study, whereas the placebo patients had 30% more at the end of the study. And again, a highly significant difference at each time point between week 6 and week 12. The medication score has changed dramatically for the placebos. What they basically did is that they substituted the corticosteroid with the short-acting beta-2 agonist. The intake of the short-acting beta-2 agonist doubled in these individuals. Here in these small graphs, you always see the absolute values. You see that the absolute values went up by a factor of 2, whereas in the actively treated group, it was actually lower throughout the entire study, with the exception of the at the last visit at week 12. The second of the clinical endpoints was objective lung function as determined by spirometry. Here you see that on placebo, the patients deteriorated rapidly when we took them off the corticosteroid here. They lost on average 250 milliliters of lung function. This corresponds to about 8%, and this is highly relevant from a clinical point of view. The actively treated patients, they remained basically with full lung capacity, which was about 3 liters at the beginning and which was the same at the end of the study, again, despite the fact that corticosteroid was withdrawn. The third of the clinical endpoints was the asthma control questionnaire, which I referred to. This is a questionnaire, as I said, which is used broadly in clinical practice. It rates 7 items which are related to life with asthma. 5 of those 7 items are patient self-reported parameters, like whether people wake up at night by asthma, whether they had limitation of daily activities, shortness of breath, etc. And 2 of the 7 items are objective items, like how much bronchodilator they use and what morning FEV1 they had. And here you see the same picture again. We had a highly significant difference overall between active and placebo. The placebos deteriorated throughout the study, whereas the actively treated ones improved. Now, there are 2 important thresholds on this asthma control scale. One threshold is 0.75 points, and this is when an individual is considered being well controlled. And this means that at this level or below, there is no need to change the treatment in these patients. The other important threshold is 1.5 points, and this is the threshold for loss of asthma control. And if somebody is above 1.5 points, it means that the risk for asthma exacerbation, hospitalization, or even death can become significant. Now, before treatment start, the patients on the corticosteroid had an average value of 0.9, which means they were above the threshold of well control. In the actively treated group, the value at the end of the study was 0.77, which means it was right on the threshold of good asthma control. If you look what happened with the placebos, you see that they went up, and they have approached with 1.49 points the threshold for loss of asthma control. Now, if we do a responder analysis and count what fraction of patients did achieve good asthma control in our study, we find the following picture. Before treatment start, only 40 and 42% in the 2 arms on the corticosteroid treatment have actually achieved the treatment goal of 0.75 points or less. When we substituted the corticosteroid with our treatment, at the end of the study, 67% of the patients have actually achieved a value of 0.75 points or less and were therefore considered as being well controlled. On the placebo, the fraction fell actually to 33%. Although we can't claim, of course, due to the study design, that our drug works better than corticosteroid, I think the data are suggestive of a very strong treatment effect with our compound, which appears to be at least in the range of the current treatment modality or even better. I would like to come now quickly to the 2 biochemical markers. We have also seen significant differences in the number of eosinophilic cells in the blood of these patients, and a non-significant, but again, trend in the amount of exhaled nitric oxygens, which reflect the same picture that we've seen in the clinical parameters. I'd like to come now to the side effects that we've seen in this study. We had in 24% of the patients injection site erythema, which means reddening, in 12% injection site pruritus, swelling, and in 6% injection site induration, edema, and pain. Basically, very similar to what you get with any vaccine, which stimulates a local or which induces a local inflammation, and this is more or less what we've had as side effects here. We have seen no systemic side effects which were different from placebo, and all the lab parameters were in the normal range. This side effect profile is supported by further 499 patients that were treated in hay fever studies with the product. The conclusions of the trial were that Q-beta G10 has shown to improve all objective and patient-reported outcome parameters in these individuals with allergic asthma. The consistent treatment effect that we've seen is compatible with an anti-inflammatory and also disease-modifying mode of action. And at the end of the trial, 2/3 of all our treated patients had their asthma well controlled, which is ultimately what you want to achieve in this therapeutic intervention. The treatment was safe and well tolerated, and we think that the product could ultimately be positioned to improve asthma control either as an add-on therapy to current treatment modalities or even as a substitute so that patients can reduce their intake of symptomatic drugs. And with this, I'm at the end of my presentation, and I would like to thank particularly the clinicians who have run the study. These are the clinical heads in 5 clinics in Germany, and our principal investigator who has done a fantastic job with this trial was Kai Michael B. from Wiesbaden. And with this, I would like to thank you for your interest.